This talk will attempt to provide an empirically plausible answer to the question: how did molecules like RNA and DNA become about the structures and functions of other molecules and the cells and bodies in which they reside? In other words, I hope to provide the first hint of how semiotic molecular information processes first arose. 2. In this talk, I want to explore some previously unrecognized parallels between genetic and linguistic evolution. They concern the question: how can the structure of a sequence of artifacts acquire aboutness, despite the lack of iconic or indexical grounding in what they refer to? In my effort to answer this, I will consider two possibilities. Can evidence from the human evolution of symbolic linguistic communication provide insight into the evolution of molecular information in biology? And can evidence from the evolution of molecular information provide insight into the human evolution of symbolic linguistic communication? 3. The underlying common connection is that both language and genetics involve displacement of reference. In 1960, the linguist Charles Hockett outlined a series of design features that he argued could be used to compare animal and human forms of communication. What he called displacement of reference characterizes the ability to communicate about things that are not present in the immediate context, including things that are absent in space or time and may even be mere abstractions. Hockett argued that displacement is a key feature that distinguishes human language from all other forms of animal communication. Displacement also characterizes the way that the genetic code is displaced from protein structure and function. 4. To begin to overcome the challenge of explaining the origins of referential displacement, it is first necessary to shift attention away from sign vehicle properties and focus on the physical process of interpretation. This is what I like to call my central dogma of semiotics. This involves 4 claims. First, nothing has the intrinsic property of being a sign. Second, anything can serve as the sign vehicle conferring reference to anything else, depending only on the structure and complexity of the interpretive process. Third, no property of a sign vehicle limits or determines its mode of reference, whether icon, index, or symbol. And fourth, the mode of reference is determined by the way that the interpretive process selectively incorporates some physical property of the sign vehicle as an affordance for mediating the relationship to something else. 5. The semiotic emergence of the capacity to utilize displaced reference to store and transmit information depends on the prior evolution of an appropriately complex interpretive competence, whether involving brains or molecules. My prior research investigated the neurological evolution that produced the competence to think and communicate with a radically displaced medium. Language. I want to explore some parallels between the semiotic evolution of language and of molecular genetics that demonstrate fundamental semiotic properties, including showing how a similar semiotic scaffolding logic underlies both. 6. Together with my colleague Joanna Ratzasek-Leonardi, who is investigating this process with respect to language acquisition, we have described this process as ungrounding. The particular focus of my presentation is on the parallels between the evolution of language and the emergence of molecular information. I will show that in both cases, the evolution of this interpretive capacity requires the evolution of linked iconic and indexical interpretive capacities to bridge between grounded and ungrounded sign media. Ungrounding enables extensive compression of information, but use of a compressed form of information is only possible because of the evolution of a complex interpretive system to decompress the information into a context that fills in what was previously compressed out. 7. Displacement addresses one of the fundamental problems of epistemology, explaining how signs can provide causally useful links to phenomena in the world, despite not being their physical cause. This has also been called the symbol grounding problem by Stephen Harnad, in which the ground is the real-world foundation on which the semiotic relation rests. There are 2 major affordances for intrinsic grounding. The first affordance is the presence of shared formal properties, or isomorphisms, between a sign vehicle and the object of its reference. This is the basis of iconicity. The second source of affordance is some physical or temporal correlation or material contiguity between a sign vehicle and the object of its reference. This is the basis of indexicality. To summarize the argument so far, referential grounding is provided by some physical property that is shared between a sign vehicle and what it is taken to refer. This property that mediates between the sign vehicle and what it is interpreted to refer to. Alternatively, referential displacement is the result of an ungrounding process. Ungrounding involves deleting or ignoring any physical properties of a sign vehicle that are shared with its referent. Maintaining reference despite this loss of intrinsic sign vehicle grounding requires referring to other iconically and indexically grounded sign relations to bridge between otherwise unrelated phenomena. 9. As a consequence, displacement of reference is supported by a kind of interpretive scaffolding. This is indirectly afforded by building on prior intrinsically grounded modes of reference. Each level of interpretation is dependent on the one below. Primary grounding is provided by recognizing some intrinsic isometry or iconic relation that is shared by some sign medium and that which is the object of its reference. Secondary grounding is provided by recognizing some intrinsic physical correlation or indexical relation by which a second sign medium is connected to both the primary iconic sign medium and the object of its reference. This makes it possible to take advantage of a third sign medium that shares none of these intrinsic sign medium properties, so long as it indirectly refers via these prior intrinsically grounded modes of reference. 10. The intrinsically grounded forms of iconic and indexical sign relations thereby provide a mediating bridge that enables the removal of all intrinsic sign vehicle affordances from a new sign medium, while maintaining an indirect semiotic link to referential ground. The resulting displacement can make it possible to take advantage of sign medium properties that would otherwise be incompatible with any intrinsic properties shared with what's represented. The removal of this semiotic constraint creates the space for utilizing iconic and indexical relationships between ungrounded sign vehicle properties and for evolving even more indirectly grounded higher-order semiotic relationships level upon level. 11. The establishment of displaced reference by prior linked iconic and indexical semiotic processes is exemplified by the familiar example of word learning in language. First, there is the iconism of repeated instances of hearing the same word sound. Second, there is the correlation between the word sound and an indicative action, like a pointing finger. Third, there is the higher order iconism between repeated indications coupled with these word sounds. In other words, a correlation between iconisms. 12. This creates an implicit injunction to discover some non-obvious iconism among the co-indexed objects of reference. This requires abduction. In other words, one must generate hypotheses about some more or less cryptic iconism that each of the indicated objects share in common. This common property is what remains after all their contrasting properties are set aside, even if all that remains is an abstract property. This exemplifies one of the most important affordances provided by referential displacement. The capacity to communicate about abstract relations is only possible due to displacement. Once sign vehicles become fully ungrounded, they provide an unlimited scope for reference that is only limited by the complexity of the interpretive process. 13. The comparison with the displacement of reference in molecular genetics is not hard to see. Displacement of genetic information is made possible by isomorphic, or iconic and correlational or indexical, intermediate molecular interactions that dissociate the sign vehicle properties of DNA from those of the protein molecules they code for. In this case, the displaced reference of DNA nucleotide codons, indicated by the three-letter groupings shown at the top of the image, is linked by isometric relations between DNA and mRNA codons and between mRNA and tRNA codons, and by the correlation of each tRNA molecule with an amino acid building block of a protein. In this way, adjacency relations between successive different tRNA molecules and their correlated amino acids represents the adjacency between DNA codons. So this multi-step scaffolding between isometric and correlational molecular relations enables the transfer of an abstract iconism from DNA structure to protein structure, despite the fact that they share no other chemical features in common. 14. This displacement enables the distinctive properties of these quite different kinds of molecules to be utilized while maintaining their contextual linkage. Proteins are directly involved in metabolic functions, whereas DNA is not. Because of this displacement between DNA and protein structures, new physical affordances are made available. For example, displacement enables proteins to recursively regulate gene expression by virtue of protein shapes shared in common with the twist of the DNA molecule. This recursive affordance enables progressively higher-order semiotic processes to be scaffolded on lower-order semiotic processes. Because of this, new higher-order functions can evolve, level upon level. 15. The molecular apparatus that accomplishes the interpretation of DNA information is a complex RNA protein structure called a ribosome. This structure is ubiquitous in all cellular life forms and is what makes the extensive hierarchic complexity of biosemiotic processes possible. In this figure, I offer just a hint of this complexity. There are, for example, 64 possible DNA-RNA codons correlated with 20 amino acids. These are listed in the box in the top left. These are paired with 57 variants of tRNA molecules, which themselves are produced by over 600 copies of tRNA genes. This is indicated by the arrow in the top middle. Each tRNA molecule has a specific amino acid attached to it by one of 37 matched amino-acyl tRNA synthetase proteins, abbreviated by ARS and shown at the top far right. These many RNA and protein molecules interact to assemble other proteins by stringing together amino acids in an order specified by the DNA and mRNA codon sequence, shown in the top right. This occurs within each of the roughly 200,000 ribosomes in each cell, like the one shown at the bottom right. Each ribosome is an RNA protein complex constructed around two very large ribosomal RNA scaffolds, shown at the bottom left. Each ribosomal RNA molecule is produced by hundreds of copies of genes for each that are widely distributed throughout the genome. These many ribosomal related RNA and protein molecules comprise the largest fraction of the mass of a cell. The high copy numbers of genes that code for these molecules is necessary to keep up with this high demand. 16. This elaborate molecular interpreting system was inherited from the last universal common ancestor of all life, known by the acronym LUCA. LUCA emerged nearly 3.5 billion years ago, resulting in a single common molecular language spoken by all known life. But all of LUCA's semiotic complexity had to have been evolved from a prior simpler ancestor, the first universal common ancestor, which we can call FUCA. This begs two questions. How did molecular semiosis first emerge in FUCA? And how did it evolve to become displaced onto molecules like DNA? 17. This interpretive competence had to evolve prior to LUCA. Without this translation machinery, the genetic code isn't information about anything. So origins of life theories can't just assume that DNA and RNA intrinsically contain information. All current origin of life theories beg the question, what is the origin of life's interpretive competence? Its informational essence. Even the most widely accepted origin of life theory, such as the RNA world hypothesis, merely accounts for RNA replication. It does not explain how an RNA molecule can embody information about other unrelated molecules and their interactions with one another. 18. So the real secret of life is not to be found in DNA or RNA. These molecules and the complex system that interprets them are consequences of a prior semiotic evolution. This forces us to ask a different question. How did the linear structure of molecules like DNA and RNA acquire the property of conveying information about the structure and function of other dissimilar molecules, like proteins and their interrelationships? 19. To approach the question of what sort of semiotic evolution preceded and gave rise to the information-bearing properties of RNA and DNA, I have proposed a molecular thought experiment that I describe as an autogenic virus. Although no such structure has been discovered or produced in a laboratory, it is an empirically testable hypothetical entity. An autogenetic virus is like a virus in structure, but unlike any currently known virus, it is non-parasitic. Current viruses must commandeer the complex interpretive apparatus of a host cell to translate its viral DNA or RNA into copies of the molecules from which additional viruses can be produced, Parasitism is necessary because typical viruses lack any molecular machinery to interpret their own RNA or DNA contents. An autogenic virus, in contrast, must generate its own components without RNA or DNA transcription, and therefore without either RNA or DNA. Instead of containing these nucleotide polymers, I envision a virus-like molecular structure that contains only two or more types of catalyst molecules that reciprocally generate each other. An example of such a simple reciprocal catalytic cycle is depicted in the image at the top left. An autogenic virus includes a collection of such interdependent catalysts contained in a virus-like protein shell. An example of a viral capsule, or capsid, is depicted at the top right. A virus capsid does not need to be assembled. It self-assembles by a process similar to crystal growth because of the geometric regularity of its component capsid molecules. A diagram of this relationship is shown in the lower right, in which proteins fit together to form the polygonal faces of a polyhedral shell. 20. The dynamical chemical relationships between these two processes is depicted by linked arrows in these two diagrams. The process of reciprocal catalysis is depicted as red arrows, and the process of capsule self-assembly is depicted as green arrows. The way that each process generates boundary conditions that supports the other is depicted as dashed arrows in the left diagram. These two self-organizing processes can become codependent when they are linked by the production of a capsule-forming molecule as a side product of reciprocal catalysis. Their codependence arises because production of this molecule promotes the growth of the capsid container, but this in turn prevents the dispersal of catalysts that depend on one another. The resulting codependence of these processes creates a higher-order molecular complex, depicted by the diagram on the right. This codependent complex can repair and regenerate itself if damaged, because damage in an environment rich in substrates available for catalysis will reinitiate both processes and thereby re-encapsulate the catalysts that both create themselves and the container that keeps them together. This codependence is, in effect, a higher-order hologenic constraint, which by maintaining the relationship between these lower-order reciprocal constraint-generating processes constitutes a higher-order self-sustaining unity. 21. In this respect, an autogenic virus is a candidate proto-life form composed only of protein-like molecules. A two-cycle sequence of autogenic damage and self-repair is depicted in this image. During such a repair process, there can be complete replacement of component molecules, while its system of codependent constraints is preserved and inherited by the newly reconstituted individual and its molecules. As a result, these constraints function as both a record of past adaptive organization and a source of possible causal influence that organizes the work that reconstitutes the whole. This also demonstrates that the autogenic analog to genetic inheritance in living organisms is the preservation and transmission of a self-reconstituting system of constraints. 22. This poses two additional questions. In what sense do these transferred constraints constitute information? And how is this information able to inform other physical processes? The short answer to both is exemplified by the way that constraints can catalyze physical processes. In chemistry, catalysts influence other chemical interactions without themselves becoming modified in the process. But a catalyst does not make the impossible possible. It only makes improbable interactions more probable. A chemical catalyst alters other chemical reactions by constraining the range of variation of the way these other molecules tend to interact with one another. This is illustrated in this cartoon of molecules interacting. Catalysts are depicted in violet, and the molecules that they influence are depicted in orange and green. Because of the astronomically many differences in their orientations, directions, velocities, and rotations, the orange and green molecules are extremely unlikely to bond with each other. Bonding will only occur when they are precisely oriented with respect to one another. However, because of the way each of these molecules is constrained by fitting into the catalyst, the probability of these two molecules binding to one another becomes far more probable. This suggests a critical relationship between constraint, catalysis, and information. Like catalysis, the power of information also derives from how constraint can make otherwise improbable interactions much more probable. In this sense, catalysis is a process that is more general than chemistry. It provides a useful analogy for demonstrating how information can influence physical events. 23. But the simple model of an autogenic virus that I have described is only composed of protein-like molecules. To complete the autogenic scenario for explaining how a molecule like RNA could acquire informational properties, I need to introduce nucleotides, the monomeric building blocks of RNA and DNA. The scenario I describe was inspired by a curious biological coincidence. Besides the informational functions of the nucleotide components of DNA and RNA molecules, nucleotides also serve as the primary vehicles for energy transfer in all living cells. Nucleotides like ATP take up and transport energy in the form of high-energy phosphates, shown as green pluses in this figure. Nucleotides have three basic parts, including phosphates. The other two are a purine or pyrimidine base, shown in purple or red, and a pentose sugar, shown in blue. With a base on one end and phosphates on the other, a nucleotide can acquire short chains of energized phosphates, from which one or more can be transferred to other molecules. The additional energy can cause changes in reactivity of the receiving molecule. In all living organisms, nucleotides are produced by protein-based catalysis, so it is not unreasonable to imagine that a system of reciprocal catalysis could produce nucleotide molecules as side products. Following a hypothesis first proposed by the physicist Freeman Dyson, I will assume that the first nucleotides were retained in evolution because of their energetic properties. 24. On the left of this slide, I diagram an autogenic reaction cycle in which nucleotides shown in green are produced as side products of catalysis. By acquiring externally available energy, shown in yellow, that can be transferred to augment other catalytic reactions, autogenic repair and replication can be greatly enhanced. But energized nucleotides can also be disruptive where chemical processes need to remain stable, as they would need to be in the inert phase of autogenesis. Freeman Dyson also suggested that by forming into linear polymers, as shown on the right, nucleotides could be stored when their energy-conveying function is not relevant or potentially disruptive. 25. But an RNA polymer can be repurposed for other functions. For example, single-stranded RNA polymers will tend to fold into branch-like 3D structures due to base-to-base bonding affinities. This is due to complementary base pair attraction between different regions of the same molecule, creating short, double-stranded branches. Non-paired regions will form loops, bumps, and hairpin-like structures connected by these branches. 26. A distinctive feature of RNA chemistry is the relationship between its nucleotide sequence and its 3D tertiary structure. Nucleotide-to-nucleotide bond affinities are diagrammed at the top left, each represented by one of 4 different colors and letters. Complementary nucleotide sequences that happen to occur between different regions along a single RNA molecule. These will tend to cause the molecule to fold onto itself to form branch-like structures where there are complementary nucleotide-to-nucleotide affinities. In this way, a specific nucleotide sequence is correlated with a specific 3D structure. 27. Interestingly, replicated RNA strands, though completely different in their nucleotide sequence, will tend to fold into the same 3D shapes. This is because replication produces an exactly complementary sequence of nucleotide bases. So where 2 segments of the molecule complement each other and tend to bind in one RNA molecule, the exact inverted sequences will also tend to bind at the corresponding region in its replicated complement. Non-annealed loops with non-complementary bases produced by complementary sequences will, however, exhibit certain different chemical properties. Specifically, non-linked loops and bumps of complementary RNA molecules will have different exposed nucleotides that are the complements of one another. So, complementary RNA replicas will have slightly varying chemical affinities despite larger-scale chemical similarities. These may increase the probability of complementary RNA molecules binding to one another. This will increase the probability that smaller RNA molecules will tend to link together to form ever larger RNA structures over the course of evolution. 28. This close relationship between the linear sequence of nucleotides in an RNA molecule and its 3D folded structure provides the possibility of a bridge between information and function. Because the sequence of RNA nucleotides determines the molecule's 3D structure, and its 3D structure determines its potential catalytic effect, the nucleotide sequence can both serve as a record of previous function and as instructions for implementing that function once again. Replication of this RNA nucleotide sequence can thereby preserve and transmit this function to future members of a lineage. Moreover, any incidental modifications of this sequence will correspond to a slight modification of function. This is the necessary condition for the possibility of genetic evolution. 29. This link between a replicable sequence and function due to molecular structure provides, in effect, a second-order catalytic function. The resulting 3D structure is effectively a template for organizing other catalytic interactions. If catalysts tend to bind to template structures in a pattern that corresponds to their probability of functionally beneficial interactions, indicated by the solid arrows in the right box, the probability of non-functional interactions, indicated by dashed lines, can be reduced. This will favor the persistence of some RNA sequences over others by virtue of how they indirectly sustain the autogenic systems of which they are a part. In other words, nucleotide sequences will be differentially retained or lost as a result of natural selection. 30. A final step in this evolutionary scenario involves offloading each RNA sequence onto a more stable, more easily replicated linear form, DNA. This is the reverse of DNA to mRNA transcription. This inverse process is currently exhibited in RNA retroviruses and retrotransposons. Although these are currently considered to be somewhat atypical processes, they may be fossils of more ancient processes. In any case, the result of this offloading is further displacement. This insulates recorded information from possible corruption by direct involvement in metabolic processes. This extra level of displacement provides a medium that is more stable and more easily replicated than RNA because its double-stranded structure is more robust and remains linear. Notice that this account of the origin of molecular semiosis inverts the logic of molecular transcription and translation as characterized by Francis Crick's famous central dogma of molecular biology. We often assume that current systems have emerged due to selection favoring their current functions. But this assumption is often misleading. Evolution proceeds by continually repurposing traits, a process that has been described as exaptation. So the function that some feature currently provides is not necessarily informative about how it originated. The evolution of semiotic displacement is a case in point where the evolutionary sequence of events and the current functional sequence of events are inverted from one another. Like the inverted logic of evolutionary displacement compared to current function in biology, a similar inversion may be relevant with respect to the evolution of language. 31. In conclusion, although this evolutionary scenario only deals with a small first evolutionary step toward the complex molecular semiosis that characterizes the protein synthesis mechanism inherited from Luca, it is the critical first step in the displacement logic that all subsequent stages in the evolution of life built upon. Standing back from these molecular details, we can now discern an analogous etiology that is shared by two quite different semiotic media, language and genes. They each evolved to record and modulate living functions while being displaced from this pragmatic ground. In both cases, displacement has enabled highly efficient compression, recording, replication, and implementation of information. In both cases, this is mediated by a medium that is functionally entangled with what it informs, as is the case with spoken language on the one hand and RNA catalysis on the other. In both cases, this entanglement links an artifact's constraint to a catalytic-like function that makes improbable relations more probable. Both have also led to the evolution of a doubly displaced, more robust medium, writing and DNA, respectively. These media are further removed from direct involvement in the functions they derive from and regulate. This allows them to be relatively inert and replicable with remarkable fidelity, and it maximizes semiotic flexibility and evolvability. This doubly displaced mode of storing and transmitting information has made possible a great many levels of cultural and biological complexification. 32. Thank you.